Good morning, children. Welcome to class 12 biology once again. Children, in a previous video, we were discussing about biotechnology, principles and processes. So, we are discussing about tools of recombinant DNA technology, and the first tools that we discussing, that is restriction enzyme. Restriction enzyme that we have discussed earlier in the previous video, but we recall some important points, then we move forward. Okay, so the restriction enzyme, that is molecular seizure, are responsible for cutting DNA. The first restriction endonuclease is Hind second was isolated by Smith, Wilcox and Kelly in 1968 from Haemophilus influenza. Okay, so the restriction enzyme recognize the specific sequence, that is palindromic sequence in DNA molecule, and they cut the DNA, okay, and form the sticky ends. Clear? After that, second tool, like you can see in the picture, mechanism of the restriction enzyme. The restriction enzyme cut both DNA at the same site, like vector DNA and the desirable DNA. Both are cut at the same site, like from G side or from G side here, they will cut and they form the sticky ends. Okay, these sticky ends you can see in the picture. So these sticky ends that are joined together by DNA ligase. Okay, so the first tools, that is restriction enzyme, which are responsible for cutting the DNA, and by this way the recombinant DNA can be formed. Okay, here the example of EcoR1, that is the restriction enzyme that cut the vector DNA and foreign DNA into the sticky ends, and these sticky ends are joined together by DNA ligase to form the recombinant DNA. Okay, after that, next tool of the rDNA technology is cloning vector. Cloning vector are form the multiple copies of DNA molecule. Okay, so vector, we can say that the DNA molecule that can carry a foreign DNA segment into a host cell, and vector may be of two types: plasmid vector and bacteriophages. Plasmid, that is autonomously replicating circular extrachromosomal DNA. Okay, so in bacteria they have their own DNA, but instead of their own DNA they also have the plasmid, that is extrachromosomal DNA. So this plasmid we isolate from the bacteria and use as vector. Okay, number 2, bacteriophages. Bacteriophages are also can use as a vector, and these are viruses which infect bacteria. Okay, so bacteriophage are the viruses that infect bacteria. Clear, children? Now their copy number, the copy number of the vector, they may be vary from cell to cell, like they can be 1 or they can be 100. So 1 to 100 copies per cell may be possible. Okay, children. The first artificial cloning vector is PBR 322 and it was developed in 1977 by Bolivar and Rodriguez. Okay, PBR 322, that is the first artificial cloning vector. They isolate this vector from E. coli plasmid. Okay, clear children? Next, now some features of the cloning vector. Okay, in this we'll discuss about the features of the vector. The first feature of the vector, that is they have the ORI site. ORI, that is origin of replication site. See in this is a DNA sequence that is responsible for initiating replication. Any piece of DNA when linked to this sequence can replicate within the host cell. Okay, so we can say that ORI, that is origin of replication site, that are present in the vector, and from here the replication begins. Okay, clear? ORI also control the copy number of linked DNA. Okay, number 2, that is selectable marker. Selectable marker helps to select the host cell which contain transformants and eliminate non-transformants. Okay, in easy words we can say that selectable marker are those substances which can recognize the recombinant DNA over the non-recombinant DNA. Okay, transformants, that is recombinant, and non-transformant, that is non-recombinant. Okay children, so if we introduce the rDNA into the host and we want to identify the rDNA, so we use the selectable marker. Okay, next, number 3, cloning sites. Okay, the various cloning sites that are present in the vector, and these cloning sites play important role in the cloning. Okay, like I'm showing a picture in front of you. Picture don't have in this slide. See without picture. So see about this cloning site. To link the foreign DNA, the vector require a very few recognition site for the restriction enzyme. Okay, like if the cloning site will present there, so the restriction enzyme easily cut the DNA molecule and it will be add to the vector DNA. Okay, so when the foreign DNA link to the cloning site, then they can easily replicate, they can easily form the multiple copies. Okay, next children, vector for cloning gene in plants and animals. Now we have seen in bacteria. After that we'll discuss also in plants and animals. In plants, basically an example, that is Anuus labus, that is TI plasmid, tumor inducing plasmid of Agrobacterium tumefaciens. Okay, actually Agrobacterium tumefaciens, a pathogen of several dicot plant. Okay, this pathogen is responsible for converting the normal cell into the tumor cell. Okay, Agrobacterium tumefaciens, that is responsible for converting normal cell into the tumor cell. Okay, because they produce a chemical, due to that the normal cell become like a tumor cell and the production will be decreased. So if we manipulate, if we alter the DNA of Agrobacterium tumefaciens and we add the desirable DNA in Agrobacterium tumefaciens, then it will lost the property of convert the normal cell into the tumor cell. Okay, फिर वो नहीं कर पाएगा. अगर हम उसका DNA चेंज करेंगे then they do not convert the normal cell into the tumor cell. Okay, so that's why retrovirus, adenovirus, papillomavirus are also used as cloning vector in animals. Okay, plants में तो that we have discussed Agrobacterium tumefaciens we alter their DNA, but in animals we use the retrovirus, adenovirus. Okay, to to prevent from cancerous cell. Okay, next. Okay, any problem regarding this, children? If any problem, you can come to school. That is very simple, very easy topic of the tools of recombinant DNA technology. In this we have discussed about, like, we recall some points. Here the tools of recombinant DNA technology that we discussing. So restriction enzyme we have discussed, restriction enzyme that are used to cut the DNA molecule. Okay, restriction enzymes are the molecular scissor which are responsible for cutting the DNA molecule. Okay, then polymerase enzyme, they help to synthesize the complementary strand. Next, that is ligase. Ligase join the sticky ends and form the recombinant DNA. Okay, clear? Ligase, DNA ligase, whenever you listen the term ligase, so that means to join the DNA fragments. Okay, vector we have discussed the two examples that we have taken about the vector. Number one, plasmid. Number two, bacteriophages. Okay, these are responsible for making the multiple copies of DNA. Okay, and vector that we have discussed, they have the following features, like they have the oocyte, they have the selectable marker, they have the cloning sites. Okay, after that, children, the next topic that we'll discuss in biotech is host organism or cell. Okay, like rDNA we have prepared till now. Now this rDNA will introduced into the host. Okay, then host show the phenotypic expression, means the phenotypic character of the host will change after introduction of rDNA, but host cannot take the foreign DNA easily. So first of all we have to increase the competency of the host. Okay, like first, that is we have to increase the competency of the host. After that it will become competent, it will be take the foreign DNA. Okay, like the two methods that we used for increasing the competency of the host, number one, chemical method, and number two, that is physical method. Okay, clear? Chemical method and physical method. The chemical method includes the heat shock treatment, and the physical method includes the micro injection method and gene gun method. Okay, that we'll discuss in detail in the next video. I hope you have learned properly the tools of recombinant DNA technology. If any problem regarding anything in this chapter, children, you can come to school and we'll discuss on board. Okay? Clear?